Break is over. So now we have to talk about biochemical properties of bacteria. Biochemical methods for the identification of bacteria. I told you that bacteria produce a lot of enzymes, and some of them are specific for some species of bacteria. So they should be detected for identification. Like catalase and oxidase enzyme, those are necessary to detect for identification. So for detection of catalase, hydrogen peroxide is used, and we have to add bacteria by inoculating loop, and then we will see bubbles and feeding through oxygen release means catalase positive. So catalase is detected by using of hydrogen peroxide. Then oxidase test. Oxidase test, oxidase is used by using of special paper strips impregnated by 13 substrates. This is paper strip placed in petri dish. It is impregnated by some substrate, and we have to add bacteria by using of inoculating loop, and we are looking for some color. And if we see the color, like purple reaction, means oxidase positive. Special impregnative paper systems are used for detection of oxidase and other enzymes. So sometimes urease, cystinase, like in case of diphtheria diagnosis, are necessary to detect. So sometimes some specific citrate utilization for border tail identification, sometimes specific enzymes should be detected. Most of the methods are based on the use of differential diagnostic environments with various indicators. Most of bacteria, pathogenic bacteria, are able to ferment sugars and to destroy proteins, especially bacteria of Enterobacteriaceae family, Enterobacteria. I told you for identification of them, it's necessary to study biochemical properties a lot of, and antigenic structure, they are biochemically active. So in case of disease caused by Enterobacteria, in case of intestinal tract infections such as typhoid fever, dysentery, cholera, and other, we have to study saccharolytic enzymes and proteolytic enzymes. How it is possible to detect saccharolytic enzymes through fermentation of carbohydrates are done by saccharolytic enzymes. How it is possible to detect saccharolytic enzymes? Because the type of enzymes bacteria produce is genetically controlled, the pattern of sugars fermented may be unique to a particular species or strain. Fermentation products are usually acid, lactic acid, acetic acid. So acid products are developed due to carbohydrate destruction, and sometimes fermentation products are acids, acid products, and gases. Carbon dioxide, hydrogen. So, as usual, acid products are developed, sometimes acid products and gases. It depends. And it's important for every species for identification of bacteria. So, the ability to ferment carbohydrates is assessed by changing the color of the medium due to the formation of acids. It is possible to detect the changes of pH. pH decreases and the indicator shows its color. For example, for detection of saccharolytic enzymes, his media were used and are used. So his media in this photo, they are a line of test tubes, not 5, maybe more, and there is a liquid medium inside, The medium very simple for enterobacteria. They don't need enriched media, at least 1% peptone water. And then in every test tube, there is certain carbohydrates such as glucose, maltose, sucrose, lactose, and so on. And there is fuchsin. Fuchsin is a dye and it has raspberry red color. But before inoculation, the medium is colorless because of fuchsin is in alkaline pH. pH is alkaline. We have to add fuchsin plus alkaline. And if the medium has alkaline pH, fuchsin is colorless. The medium has slightly yellow color only. And inside the medium, there is inverted tube. And inverted tube is like small test tube, and it should be placed in the liquid medium bottom up, and the medium should be inside. This inverted tube is used for detection of gas production. So his media before inoculation has no any color. And now after inoculation of specimen, or when these media are incubated at thermostat, then we are looking for the color. In this photo, you see that, это что-то все не то. For this picture, so after incubation in thermostat, now we see color of fuchsin. And by the way, there are biochemical properties of Escherichia coli that you will study it. And there are test tubes with carbohydrates such as glucose, lactose, maltose, mannitol, sucrose. And now we see color of fuchsin, and we see bubble of air in inverted tube. So the conclusion is made that Escherichia coli are able to ferment glucose, lactose, maltose, and mannitol till acid and gas production. It's important to write the results exactly what products are developed. So in this case, we see acid products production and gas production. The only test tube, the way medium did not change the color, is the medium with sucrose. It's still the same and there are no any gases. So Escherichia coli are not able to ferment sucrose. They have no such enzyme. They are sucrose negative. And then proteolytic enzymes. How it is possible? So a bright yellow color indicates protein cleavage, detection of proteolytic enzymes. How it is possible to detect proteolytic enzymes? Proteolytic enzymes are detected by indole and H2S production after inoculation of bacteria in liquid medium with peptone broth. Because of peptone is the product of proteins destruction, and if bacteria have proteolytic enzymes, bacteria those are inoculated in this broth, they are able to destroy peptone till amino acids with gases production. Really 3 gases are produced, but in microbiology, we are going to detect just 2 gases, indole and H2S. By using of special gases go out from the liquid medium, and for detection of them, well, this is picture of the test tube with 1% peptone water, and special strips, papers impregnated by 13 chemicals are placed between the cotton stopper and the wall of test tube, 2 papers. And if bacteria destroy the proteins, gases go out, and due to reaction with specific chemicals, we will see changing of color of papers. If the paper has a red color, means indole production, and if another paper will get black color, means H2S production. So here on this photo, indole is produced, H2S is not produced. They are properties of Escherichia coli. Really not necessary to learn by heart all these properties, but for Escherichia, you have to memorize that they are able to ferment glucose and lactose, and they are sucrose negative because of Enterobacteria family bacteria are divided on lactose positive and lactose negative due to lactose fermentation. And you're not going to study all of them. Just memorize that Escherichia are lactose positive bacteria, and Salmonella and Shigella genera bacteria are lactose negative bacteria. So for primary inoculation of feces, and of course, these bacteria are able to cause intestinal tract infections, and they go out by feces. And for isolation of these bacteria, feces should be inoculated on solid media, differential media used. Those content of agar, nutrient agar, plus lactose, and plus indicator plus alkaline. Like, for example, McConkie medium. And on McConkie medium, it is possible to differentiate lactose positive bacteria from lactose negative due to lactose fermentation because of Escherichia bacteria. Those are able to ferment lactose, and due to lactose fermentation, pH will be changed, and indicators that was colorless before inoculation because of alkaline pH will develop the color, and the colonies of lactose positive bacteria, Escherichia, will get the color of indicator. They will get red color on McConkie medium, and other bacteria of Salmonella genus and Shigella genus, they are not able to ferment lactose. They are lactose negative, so the pH is not changed, and they will develop colonies. Anyway, they will multiply, but without changing of pH, and they will develop colorless colonies. So there are many differential media. Indicators are different, dyes are different. Anyway, Escherichia will get color of indicator, maybe color of fuchsin, raspberry red, maybe blue color if methylene blue is used, and so on. And colonies with the color of indicator are formed by lactose positive bacteria. Colonies without the color of indicator are formed by lactose negative bacteria. We will study Salmonella and Shigella, so memorize, it's very important. Then his media were developed last century, and they were used very widely during many decades. Now they are used, but nowadays the modern methods are developed to identify the bacteria. Also are used paper indicator systems. I did show you paper indicator systems. Maybe they are looking like paper discs impregnated by 13 substrates and indicators, and in this case we should place every paper disc in test tube. Pure culture should be inoculated on liquid media, or maybe we have to use polystyrol plates with dippings, and we have to place these paper discs, and again by changing the color we will see the presence of some enzyme because of changing of pH. And I did maybe paper paper strips, like here I did show you paper strips impregnated by some substrate and indicator, and again because of changing of color it is possible to detect enzyme. Nowadays polystyrol plates are used, named microtests. They are plastic plates with various substrates and indicators in the wells, in the dippings, and dilutions of bacteria are introduced into the wells and incubated at 37 degrees by Celsius at thermostat. And then again, due to fermentation of substrates, we will see changing of color, and it is possible to see results in 4 of 8 hours. And due to instruction, it's possible to make conclusion, is result positive or negative. They are one time you're using plates, and they are industrial, they are commercial, they are produced, and very often such multi-micro tests are used for identification of Enterobacteriaceae family. Enterotest 1, Enterotest 2, and different substrates are absorbed in dippings of these polystyrol plates. So it is possible to detect many enzymes. Nowadays, automatic systems are developed, and the macroscale system is a complex of plastic plates containing various substrates, and gram-positive and gram-negative bacteria differentiate by fluorescent substrates, and the analysis times is approximately 2 hours. Then systems with egg plates with 30 holes is used. A suspension of bacteria is automatically introduced, and analysis time from 6 till 18 hours. So different kinds of plates are used for detection, and here it is written what is the result. Urea, citrate, and arabinose, sorbitol, lysine, and so glucose, and bacteria are differ. There are a picture of exactly bacteria, those belong to Enterobacteriaceae family, like Proteus genus bacteria and their results, then Enterobacter elegans, Escherichia coli, and maybe Hismedia, maybe some radipolystyrol strips are used, and nowadays it's possible to place the polystyrol plates in special equipment. Special systems are computerized and automatically perform all calculations. What we have to do, we should isolate pure culture, and then should be placed in biochemical analyzers, and biochemical properties are detected by this equipment automatically, and then the information goes to computer, and the even identification of bacteria is done automatically. So this is a modern equipment. Have you any questions?
No.
Then now you should go to the education. Then you will study more about physiology of bacteria on laboratory classes. And now you should go to educational portal. Did you answer the test for the previous lectures?
Excuse me, doctor.
The leaders of group,
group,
I'm repeating, inform students that finally the attendance of the lectures will be checked exactly due to answers for test, answering for test. The tests are opened after the lecture during the day till 7 p.m. Then the test will be closed, and then the attendance of lecture will be checked by answering of tests. And every hour of missed lectures will be placed in finally in computer program for a calculation of your final rating. All hours of missed laboratory classes, all hours of missed lectures. Of course, student anyway will do the work of missed class or missed lecture at the end of December. I will open all tests to do the works anyway. Even student will do the work. Anyway, all these hours of missed lectures will be calculated by computer program. So inform that you should go now on educational portal. I know that you are going for class, so I did finish lecture before, and I gave you time for answering for test. You have no excuses, and then I will close the test. So the leaders, you're responsible. Could you inform all students, please? Do you hear me? Did you find the educational portal? It is written microbiology, pharmaceutical faculty, and every in accordance with your group number. Don't mix group number. You should answer for tests. And then I will place presentation. I am repeating again that the presentations of lectures are placed on educational portal, so you have opportunity to prepare for laboratory class in accordance with lectures. Use the lecture, and after the lecture, I will play the video lecture on educational portal with my comment. Use the lecture, use the textbook. The textbook is available for every student. If you don't know how to use it, go to the library. On the second floor, there is internet room, and every student should come personally, and the librarian staff will help you to get password personal, to get the electron textbooks and manuals. Don't try to find excuses, find opportunities. Okay. Have you any questions?
The test will be open till what o'clock?
Till 7 p.m. today.
Okay, thank you.
Welcome. So if you have no questions now, answer the test, please. Goodbye. Next lecture will be in 2 weeks from now. The next lecture will be in 2 weeks from now. And I've sent you information. It should be 7th of March. Нет, это не будет. В четверг же у нас. 24th, then on 10th of March, the next lecture on 10th of March. I've sent you plans of lecture with dates. Goodbye.
Okay, thanks, Doctor.
Goodbye.
Goodbye.
Bye-bye.